my talk at ADPD focused on a very important aspect of Alzheimer's pathology, which is also somehow understudied, even though it's becoming more and more recognized, which is neurovascular dysfunction. In particular, we are looking at microvascular endothelial cell dysfunction in relation to Alzheimer's disease and cerebral amyloid angiopathy. So cerebral amyloid angiopathy is the deposition of amyloid around the cerebral vessels, which is very important in the disease pathology because, first of all, it's present in almost like 90% of Alzheimer's disease cases, and also in a lot of aging brains without Alzheimer's. So it's a very common pathological feature. It is synergistic with cardiovascular risk factors. So if you have problems in your cerebral vessels, you probably have more difficulty in clearing the amyloid from the vessels. So the amyloid further accumulates there and it creates more problems. So something that we are starting to study now is actually the potentiation of the effects of CAA by cardiovascular risk factors. And of course, it's something that we don't know that much about the molecular mechanisms that are involved in. So we want to study more what happens, right, when amyloid attaches to basically the vessels, what happens to endothelial cells in these vessels. CAA can be in big vessels, but also in capillaries and most small vessels. So we are really looking at the mechanisms of these functions. So one thing, my talk was basically divided in two main parts. The first part is a study that we published recently, which was focused on the effect of different fragments and types of peptides that are present in the Alzheimer's brain, in particular on different aspects of endothelial cell function and viability. So we looked at how peptides that are very short, they are cleaved form of amyloids and don't aggregate, or longer peptides, so the classical A-beta-40, 42, or vasculotropic peptides, so peptides that are very related to cerebral amyloid angiopathy, such as the Dutch mutant, or even forms that are cleaved at position 4, like the 4-40, 4-42, which are known to aggregate more and form a lot of fibrils. So how these different peptides basically affect endothelial cell function. And what was very interesting is that we found different reactions based on what we looked at. So if we looked at apoptosis, we saw clearly that the oligomers, the peptides that form oligomers, induced apoptosis, while peptides that remain monomers do not, and peptides that immediately aggregate to form fibrils also don't cause apoptosis. But if we looked at blood-brain barrier permeability, and we looked at that placing the endothelial cells on electrodes and measuring with a system that is called the ECZ-theta, which measures in real time blood-brain barrier resistance, basically endothelial barrier resistance. So in that case, the fibrils were a lot more toxic. They decreased blood-brain barrier resistance more than the oligomers, and the monomers didn't do anything. So there was like a different reaction, right, in the two outputs. And then we looked at angiogenesis. When we looked at angiogenesis, all the peptides, even the monomeric ones, were affecting somehow angiogenesis, decreasing the ability of endothelial cells to form vessels in matrigel. But the more the peptide was aggregation prone, the more angiogenesis was destroyed. And in this case, also like in blood-brain barrier permeability, fibrils were the worst, like the ones that affected the most. So this was the first part of the study, which is really interesting because it connected well to many of the talks of our session in which I were talking about different species of amyloid present in the brain. The other part of the study is more related to the mitochondrial mechanisms. So we saw, and we previously published, that endothelial cell apoptosis induced by amyloid is mediated by, first of all, interaction with death receptors on the cell membrane. And then those death receptors trigger a signaling pathways that goes to the mitochondria and induces mitochondrial cytochrome C release and production of ROS, reactive oxygen species. These was published already, and we were looking for compounds, for drugs that could block these mitochondrial dysfunction mechanisms. So we went through, actually someone did the groundwork. They went through 1,000 drugs of the NINDS drug bank, and they found a few that were active at inhibiting cytochrome C release, previously already published. So we took some of those, and we found that the one that worked the best in our model was methazolamide, which is a carbonic anhydrase inhibitor. We studied methazolamide and acetazolamide, which is an analog that is very well known, used, for example, for glaucoma, high altitude sickness, in our models of cerebral amyloid angiopathy. And very interestingly, we saw that they inhibit mitochondrial dysfunction, in particular loss of mitochondrial membrane potential, production of hydrogen peroxide, but also when we tested them in animals, they were able to reduce amyloid pathology, reduce cognitive impairment, and also we are looking at different pathways related to inflammation mechanisms because they reduce gliosis, they change the response of microglia. So we're still working on these studies, and it's all very exciting. And